Today we're going to begin our video on the urinary system. To begin with, our urinary system consists of only 4 primary organs, therefore types of organs. There are 2 kidneys, 2 ureters, a single urinary bladder, and a single urethra that leads out of that. So our kidneys up here produce urine, travels down through the ureters on either side, enters into the urinary bladder from below, not from above, but from below. We have these 2 openings in the wall of the urinary bladder down here where urine enters into the bladder from either ureter, and then finally the single urethra that leads out of the urinary bladder. It's fairly short in females, and when we look at the male, we'll see it's longer and divided into 3 different segments of the urethra. Next, we're going to look at the positioning of these organs inside the body cavity in a couple of models. In this model, you see the 2 kidneys. They are here about 2.5 to 3 inches in height. At the top of the kidney, you have the adrenal gland, sometimes called the suprarenal gland, up above the kidney. The ureter leading out of the kidney, here on this side and here on this side, loops over the front of the iliac artery near where the common iliac branches into internal and external iliac arteries here. That ureter stays against the wall right back here and then stays against the wall on the inside of the pelvic cavity all the way down to the lower floor of the urinary bladder. In this model, you see the same thing. This model is a little bit larger. Here's the kidney on this side, the kidney here, and the ureter is coming down on either side. The adrenal gland or suprarenal gland here. Notice the proximity to the spleen. They show the spleen right in here. This would all lie just a little bit behind the stomach. About right here would be the stomach, and the pancreas would also come across right about there. These ureters, remember, stay against the wall on the pelvic cavity and come into the bladder from below, so they're not suspended out in the body cavity. Next, we're going to do a look at the inside of a single kidney to see the kidney anatomy. All right, now we're going to look at the inside of the kidney. The kidney, if you cut through it from top to bottom, it's kind of what we consider a frontal section, so you separate the front from the back. And look inside, we can identify some specific regions and parts we want to label in this diagram here. First of all, on the very outside, there's a connective tissue capsule on the outside of the kidney. So the outer capsule. Beneath the capsule, there's a zone of tissues that look a little bit different than the next zone in. This is called the cortex. Here, we'll talk more about the details of structures in the cortex in a minute. The next zone in here, let's see, I can put a little bracket on here so you can see its distribution a little better. This is the medulla between the cortex and what we call the renal pelvis to the inside. So this right here is the renal pelvis. So we've seen renal cortex, renal medulla, renal pelvis. Branches of this pelvis go out in different directions are referred to as calyces, and we're going to identify some calyces here. The larger ones, the first branches here, these are called major calyces. This is a major calyx, C-A-L-Y-X. And then these smaller ones out here would be minor calyces. So I'll label this one right here, a minor calyx. Minor calyx. The general region right here where that renal pelvis is connected to the ureter is known as a hilus. And at the hilus, we have the artery coming in and the vein going out. So there's an artery that comes in here. Directly off of the main aorta. So the aortic blood supplies that renal artery. And then there's a vein that comes back out the other way, carrying blood that's already been through. And this goes to the vena cava, the inferior vena cava. So it's a fairly direct blood supply route from the aorta to kidney, and then from kidney to vena cava. Okay. So anyway, like I said, I'll make a dotted line right here to represent kind of a bracket around that region. It's called the hilus. We didn't identify the ureter here. And I'll just do the renal artery and vein. Renal A and B, we'll say artery and vein, renal artery and vein. Another term we'll sometimes run into is renal sinus, the connective tissues that are between the different branches of arteries and the calyces in here before we get into the medulla. That loose tissue surrounding those parts, sometimes referred to as the renal sinus. Renal sinus. Another important thing I forgot to mention as I was going through here is that the renal medulla that we have here is divided into segments, more or less, sectors, I guess would be a better term. Sectors that drain into these minor calyces. And those sectors of the medulla that drain into the minor calyces are referred to as renal pyramids. There's a single renal pyramid there. So we're going to look in detail at nephrons here in a bit, but nephrons produce urine. Urine moves into these collecting ducts indicated by these lines that I've drawn in the medulla here. Those lines, those ducts, drain the urine fluid into the minor calyces. And then it moves from minor calyx to major calyx and then to the center of the renal pelvis here. Here we have a fairly high quality model of the kidney. On the outside we have the capsule, and then we have this zone. We can see this band around the outside that is the cortex. The medulla made of medullary pyramids all around here. Minor calyces where the pyramids come to a point and drain urine fluid into the minor calyces here. So there's minor calyces. This would be a major calyx. There's a major calyx. There's a major calyx. This would be renal pelvis here. Here's the renal artery coming in. It branches out and feeds blood out to the perimeter. And then the veins bring the blood back. And this is the renal vein. This region here was the hilus, sometimes called hilum or hilus. And these kind of open spaces between things in here would represent regions of the renal sinus, And this is the ureter coming out of the renal pelvis right here. In this same model, we see a magnified view of what's going on here in the cortex and the medulla to form nephrons and collecting ducts. So in this section here, we have these little light colored balls. These represent renal corpuscles, and we'll talk about those here in a minute. And from the renal corpuscle, there are little tubules that extend out. These are parts of the nephron. All these are parts of the nephron. Here's a loop, the loop of Henle of the nephron, coming back up to more twisted, what we call convoluted tubules of the nephron, and then it drains into a collecting duct, and then our collecting ducts find their way through the medulla and down to the calyces. We are going to next look at the overall structure of a single nephron and collecting duct. Now we're looking at a drawing of the nephron structure. All of this through here, this twisted tube going down and back up all the way over to here, this is all nephron. Here is where the nephron meets a collecting duct. So there would be several nephrons connected to one collecting duct. So all of these are openings to the distal tubules of other nephrons. Let's go ahead and talk about what's going on here with this structure. First of all, there's a blood supply to a capillary network here. That blood supply right here, we're going to label as the afferent, A-F-F-E-R-E-N-T, arteriole. The afferent arteriole goes into this capillary network. That capillary network there is called a glomerulus. After the blood moves through the capillaries of that mass of capillaries, sometimes called a tuft of capillaries, that mass of capillaries, it moves into another blood vessel. It's called the efferent, or efferent, E-F-F-E-R-E-N-T, arteriole. And this is where filtration of the blood plasma occurs. So some of the blood plasma fluid will move from the blood out into a space around here we call Bowman's capsule. Bowman's capsule. Bowman's capsule. That fluid, after it moves into here, is then pushed into this section of tubule. That tubule region here is referred to as the proximal convoluted tubule. This is in the cortex region of the kidney, the outer portion here. From here down would be in the medulla of the kidney. So let me just kind of put a dotted line across here, indicate this is cortex out here, medulla down here. So your proximal convoluted tubule, the fluid moves through here. Along the pathway, many of the molecules that your body needs to retain and not lose in the urine are retrieved here. All of your glucose is supposed to be retrieved from your blood here. Many of your ions, important ions to body functions, are retrieved here. And along with that, some of your water is removed. Water moves along as your solutes are pulled out. Water will follow osmotically. Eventually, the fluid moves into this loop we call the loop of Henle. Loop of Henle goes down into the medulla region and then back up again into the cortex. After it comes up into the cortex, a part of that nephron tubule then comes up right adjacent to the connection of the glomerulus there. But this entire section is called the distal convoluted tubule. Distal convoluted tubules are not quite as long as the proximal convoluted tubule portions, but anyway, they lead eventually out into the collecting duct. Earlier I used the term renal corpuscle. Renal corpuscle refers to this entire mass here, that spherical structure, and it includes both the glomerulus and Bowman's capsule. So all that together makes up a renal corpuscle. Renal corpuscle. Glomerulus and Bowman's capsule. So talking again about what is going on here exactly, remember we have blood supplied here, we have filtration of the blood, we have a lot of fluid added to this space called Bowman's capsule. There's a large volume of your blood fluid that moves through there and into all of the different Bowman's capsule spaces in the kidney. So most of that water, that fluid, needs to be taken back into the body, retained within the body. So there are some things we want to get rid of, our kidneys are trying to excrete or get rid of. We want those things to stay within the fluid all the way out to become part of the urine. Other things your body wants to retain. So we have a lot of the retention of good stuff, things you want to retain, in the proximal convoluted tubule, and a lot of our water comes with that. The loop of Henle is specifically adapted to pull water out of the urine. So in this portion of the loop of Henle here, we're pulling a lot of solutes, a lot of sodium and chloride ions moving out of the loop of Bowman's, loop of Henle, rather. And as we add more solutes on the outside, we bring more fluids along with it. So our water retention, a large part of it happens here in the loop of Henle, in the medulla portion of our kidney. Eventually the fluid moves on out. We still are going to be pulling out a few things that we want to retain in our distal tubule. We'll also be excreting a few molecules that we still want to get rid of with some of these cells in the distal tubule. And eventually that fluid, the urine fluid, moves into the collecting duct, and the fluid from the collecting duct moves on down. And even in the collecting duct, in the medulla portion, we can pull some more water out, retain water, not lose too much water with the urine. So that's the anatomy, the overall anatomy of a nephron. This is something that I expect everybody to learn and know how to label all parts of a nephron, and know to some extent where the fluid movement is and what's going on with that. So our afferent arteriole has blood moving up and into the glomerulus. Efferent arteriole, the blood is moving out to circulate still within the kidney, do some more circulating before it's returned to the renal veins. I wanted to come back to this model just to have another way of looking at the nephron. Remember that these little white spheres we have here represent the outside of the Bowman's capsule. The whole thing would be a renal corpuscle. So this renal corpuscle right here is cut open so we can see the vessels of the glomerulus inside there. So we have the blood moving in and then back out again. But anyway, there's our opened renal corpuscles there, showing the glomerulus inside of Bowman's capsule. So that white circle around it is the Bowman's capsule. Then we have the proximal convoluted tubule portion leading into a loop of Henle that goes down and back up. And then we have the distal convoluted portion here, and then coming into the collecting duct. Another on this side, this is proximal tubule, loop of Henle, then the distal convoluted tubule leading into the collecting duct. And you can see different nephrons draining in along the way down the collecting duct right here. And of course that ends up at the minor calyx of the renal pelvis. Another thing we want to see here, this is a magnified view of a single renal corpuscle. The afferent arteriole coming in, and these are the loops of the capillaries within the glomerulus here, and then an efferent arteriole leading out. This is the space of Bowman's capsule here, and then the fluid moves from Bowman's capsule into the proximal convoluted tubule right here. So that's the first part of the proximal tubule. It goes out, twists around, goes down through the loop of Henle, back up, and then this part right here is the distal convoluted tubule where it passes next to the connection of the glomerulus. So we've already dealt with the kidney in some detail and talked about the structure of the kidney, how it works. The ureters convey the urine from the kidney to the urinary bladder. There's not much detail in the structure of the ureter. They do have some smooth muscle in the wall, and they do have some peristalsis, like contractions that travel down the ureters. The inner lining of the ureter is that transitional epithelium that can be stretched and distorted in shape to allow some expansion of the ureters. And they lie behind the body cavity. They're not in the body cavity. So on their front face, there will be that peritoneal lining, but on the back, there'll just be adventitial connective tissues that join them to adjacent structures, muscles and other structures in the abdominal and pelvic cavity. So those two ureters come down and they enter into the urinary bladder here on the floor. They stayed outside of the body cavity and came in underneath the bladder here. At this location, these two openings right here and the exit of the urethra right here, these positions remain fairly stable, making a region on the floor of the bladder that doesn't experience a lot of distension and contraction. So it's a kind of stable structural region known as the trigone. The trigone. Trigone. The lining of the bladder consists of that layer of transitional epithelium. Beneath that, a little bit of connective tissue indicated by the green here, and then the muscularis layer, muscularis externa, of the urinary bladder to the outside of that. That's all smooth muscle, and unlike parts of our intestine or esophagus or stomach, where that muscle is all running in specific directions in any particular tissue layer, in the urinary bladder it seems to run in a lot of different directions, and mixed between that smooth muscle is a fair amount of connective tissue as well. So we have muscle and connective tissue kind of mixed up in there, and then when you get to the outside of that, we have a thin layer of connective tissue in there just underneath of the peritoneal lining, that mesothelium of the peritoneal lining, indicated in brown, connective tissue there in green underneath of it. So on the outside we have a serosal membrane. So the layers in this, there's a mucosa. It consists of the transitional epithelium and the underlying lamina propria. So the transitional epithelium and then lamina propria. Next, we have a muscularis layer. It's all smooth muscle, and it's mixed with a fair amount of connective tissue. And then on the outside of that, we have that serosa, a mesothelium, and then underneath of that, a little bit of connective tissue there. You'll notice that that mesothelial covering in the peritoneal lining does not cover the entire bladder, only the upper part of the bladder. This lower part of the bladder against the floor does not have a mesothelial covering. Its connective tissues would lie adjacent to other structures down here. So these would be adventitial coverings down here, not serosal coverings. Now at the very bottom of the bladder, that muscle layer that would run every which direction around most of the bladder, comes to a point here where there's an inner circular layer I'll label that circular smooth muscle. This is involuntary muscle, but it forms a sphincter muscle at the very lower end of your bladder, right at the very top, I should say, of the urethra. There is that circular smooth muscle forming a sphincter, the urinary sphincter, the internal urinary sphincter. Outside of that, beyond that, we have skeletal muscle of the body wall. So this skeletal muscle makes a second sphincter, and this is voluntary muscle. So we have an involuntary inner circular muscle and a voluntary outer skeletal muscle sphincter at the upper part of the urethra, right here.